So, okay, one of the questions, if you don't know this, I'm sorry, you just did a quick look, but it was like, what did Nathan Radley claim?
Okay, so here's how you want to look at chapter 8, okay? So I kind of bracketed stuff. So if you kind of take all the, when you get ready to study for something like biology, you got to take all that information and figure out how do I organize this so I can get it to stay in my brain, right? And so these two bullets go together, right? ATP and this one are together, and then the rest of this is all enzyme stuff, okay? So if you look at chapter 8, what will most of the questions very likely, what are they going to be over? Enzyme stuff, right? You're going to have a handful of these, but this is the meat of the chapter, okay? So I don't know if that helps or not, but just pointing that out. Okay, ready? So let's kind of put all these words together. Catabolic means what? To break down, all right? Catabolic goes with being exergonic. Catabolic and exergonic go with hydrolysis, okay? And then if you want to tie these in, okay, since it's catabolic, exergonic, hydrolysis, let's talk about delta G, okay? That's going to be, you want a table?
Yeah.
Like a T-chart kind of thing? All right, let's do that. All right, so we've got build and break. Okay, so let's do the breakdown stuff. Okay, so we said catabolic. Catabolic is exergonic. Okay, it's hydrolysis. Negative delta G. Okay. So we're releasing energy. Spawn, non-spawn. Spawn. Because it's, if I graphed it, overall, the graph is going down. Energy is released. Okay. So now let's do Bill. Where someone was right there. Let's do building. So what's building? Anna. Hold on, mess that up. Ender. Ender, endo, it doesn't matter. And it's dehydration synthesis. It is a positive delta G. And it is non-spawn. And then if I graphed it, overall it is uphill. Okay? So then if you look at the two processes, we've covered one of them, respiration and photosynthesis. Out of those two, which one represents building? Overall. PS, right? Photosynthesis. And then this is RS. Okay? Respiration. Cellular respiration. Right. Okay, so we just knocked out a lot of stuff right there. Feel good on that? And there are some just questions like that. So you should, just don't get, the two I see you guys get mixed up are these two right here. So do you have a way to remember cata and anabolic? Anabolic steroids getting swole, right? Catabolic, we're breaking down the cat. No? Yeah.
I don't like that. Yeah.
Oh, random fact. So cats like to wake you up in the middle of the night for some reason, right? I read an article about it. and they found that cats are just checking to make sure you're okay. Yeah, they look at you as an awkward cat. That's how they look at you, and they're just checking on you to make sure you're still alive and everything's good. So yeah, it's kind of weird, but that's a cat. Okay, so let's look at these. All right, so we have delta S, delta H, delta G. Okay, the one you always want to start with when there's one question about this. What does delta S stand for? Change in entropy, right? Entropy. This, and it's not really that you have to know much about enthalpy. And then this is change in free. Okay, so if we are breaking down, okay, we're breaking something down, okay, so we're taking this and we're turning it into that, okay, we're breaking those bonds. What are these going to look like? What's going to happen to entropy if I break something apart? More or less? More disorder, less disorder? More disorder if it's breaking down, okay? Then with these two, because I'm breaking the bonds, you're losing the heat and you're releasing the energy, right? Because again, free energy downhill is negative delta G. We're breaking stuff apart, that's negative. Okay, so if you know breaking down, you know building up. Because building up would be, this would be negative, and these would be positive. So this column is build, this column is break. See it? Okay. And as long as you understand entropy, you can figure out the other ones, because everything is opposite of what entropy is, right? Entropy is a measure of disorder, right? And so again, you cleaned your room maybe yesterday. By the end of this week, it's going to be messy unless you're fanatical about keeping your room clean. You know, some people are. So. Okay. All right, so that's this one, what occurs. So synthesis and digestion. Got it? Okay, can I take that away? All right, ATP. So ATP, adenosine triphosphate, and what matters are the 3 P's, PPP, right? Because this is the, I'm struggling with spelling today. Okay, so there's our adenosine, and there's our 3 P's, okay? Which phosphate is most unstable and comes off? The last one, okay? So the last one comes off when that's, so are we dehydration synthesizing, or are we hydrolysis? Hydrolysis, right? Hydrolysis, we're taking that phosphate off, this last one, and when that comes off, energy is released, okay? But that's the structure of ATP. 3 phosphates, adenosine, you take phosphate off, energy is released. Got it? Okay, and then when ATP loses its phosphate, it becomes ADP, right? There's no AMP stuff, even though you saw that. Just ADP and ATP, okay? Okay, feel good on that section. Clear? All right, ready for enzymes? Okay. Okay. What's what? Okay, so we've got the substrates, and here is active sites, right? Enzymes are substrate specific, meaning they they're very specific to what they react with, right? They're substrate specific. So there's an enzyme for everything, right? You can't use an enzyme to break down lactose and turn around and use it to break down proteins. There's a different enzyme for breaking up proteins, right? Okay. So enzymes, purpose and structure. So there's the structure, the purpose, okay? I'm gonna hold off on the purpose because we're gonna do it right here, okay? But overall, enzymes are defined as they lower the activation energy. And I'll talk more about activation energy in a little bit, but that's what they do. They speed up reactions by lowering the activation energy, okay? So factors that affect an enzyme. So we'll lump these together. They all kind of go with the same thing. So what is this peak called? Optimal. Right, optimum, right? So optimum meaning the best. So if we're talking pH, what is the optimal pH of the majority of enzymes in our body? 7, okay? If it gets too basic or too acidic, enzymes are proteins, so they denature, they fall apart, okay? That's optimal pH. I can do the same thing with temperature. Celsius, that's 98.6, that's 37, right? So pH, the temperature of enzymes that's the optimal. All right, so there's two differences here though. If it gets too warm, heat does what to the bonds of an enzyme? Denatures it. Whereas cold slows it down, kind of like your hands when they're cold and you have to do something with them, right? It slows them down. So cold slows them down, heat denatures them, okay? Salinity is, there's not really a number, but there's a salt concentration, there's an optimal salt, okay? So all those things have the optimal, the best conditions. These are all factors that can affect enzymes, okay? So if I go back to this, if I put something right here, that red thing is blocking the substrate, what's that called? That's a competitive inhibitor, right? It's competing with the active site. If I take this enzyme and this red thing binds, which changes the active site shape, that's called a non-competitive, which can also be called allosteric. Non-competitive and allosteric go together. Okay, remember allo means other, homeland, other, this is the other region, okay? And then feedback inhibition also deals with allosteric. We'll hold off on that for a second. Okay, cofactors, coenzymes, okay? So, which ones are vitamins? Coenzymes. Okay, these are vitamins, they are helpers. Okay, coenzymes are vitamins, they help the enzymes work better. That's why they say if you take a multivitamin, it helps you. Okay, those vitamins are good for you, they help your enzymes work better. Cofactors, another one is, these are organic. Okay, cofactors, they are inorganic things, okay? Like, they're not vitamins. It could be iron.
Are they still helpers though?
They don't really help, they're just part of the enzyme. Like, hemoglobin has iron in it. That's a cofactor, not a coenzyme, okay? The difference though, how do you know the difference? Inorganic, organic. Vitamins, not vitamins, okay? That's one single question anyhow, so. Okay, all right, then feedback inhibition. All right, so if we have A, B, C, D, okay, and then we have arrows, what do the letters represent? Substrate. The letters are the substrates. Okay, the arrows are the what? The enzymes. Okay, so this assembly line has 3 enzymes. Okay, so we convert A to B, B to C, C to D. Okay, but let's say we are accumulating excess amounts of D. Okay, well, your body's an efficient machine, so we need to shut this pathway off temporarily. Okay, so let's say this is enzyme 1. Okay, this is substrate D. Where on here would substrate D be? It's a non-competitive, right there. Okay, so it's allosteric, right? So allosteric is feedback inhibition. That's allosteric. And because D binds to enzyme 1, what did it do to it? It shut it down, right? It altered the active site shape. The enzyme is temporarily switched off until what? All D is gone. And then once all D is gone, what happens right here? It goes away, and now the enzyme's been switched back on. Okay, we're back in business. Okay? That whole process is called what? Feedback inhibition. Got it? Okay. So those are all the things that affect an enzyme. Good on those? All right. Can I clear it? Okay. Why do enzymes plateau? Okay, so and you guys saw this relationship in your toothpick lab. Okay, all enzymes do this, and then they plateau. Okay, even if I add more enzymes, I'm gonna add more enzymes. Okay, well, it still does this, but it still plateaus. If I add more substrate, what's it gonna do? It still does the same thing. It increases the rate of reaction, but it always does what? It always plateaus. Okay? So again, think about it. If you have all these substrates that we're trying to break apart, okay, and we have all these enzymes, okay, why, looking at this picture of what I drew, why is it plateauing? The active sites are occupied. They can only work so fast. That's exactly it. And so, yeah, for a while it'll speed up the reaction, but it'll always plateau because the enzymes are busy doing their job, right? Their active sites are occupied. As soon as they finish it and release them, then they're going to go attach to more, but it's still the plateau effect. Got it? Okay, so that's why. Okay, next is the graph. So you know you're going to have an enzyme graph, right? Not to make, but to interpret. Okay. So let's label all this stuff. Where, if I had to draw activation energy, where would it be? Okay, so here's one. Is there another one? Okay, which one represents with the enzyme, which one represents without?
The black one is with
Okay,
enzyme.
so this is with enzyme, and this is without enzyme, right? So these red lines represent the E of A. What's E of A stand for? EA Sports? Activation energy, right? So let's go back to the definition of an enzyme, okay? Enzymes lower the activation energy, doing what to the reaction? Speeding it up. Lower the activation energy, thus speeding it up, right? So let's say you had 5 guys yesterday with some French fries, right? You digested that quickly because you have enzymes, right? Because you have enzymes, it lowered the activation energy, it sped it up, you broke down your double 5 Guys burger. I like 5 Guys with the onions and mushrooms on it. Good stuff. Okay, so where's delta G at? Right there. This area right here is delta G, okay? Regardless of whether or not we have an enzyme, does delta G change? Delta G stays the same, it's just with an enzyme, you release the energy faster, right? Again, you don't change the delta G, you just get to it faster, you release the energy faster. Okay, by the way, positive or negative? Negative, right, okay? So that's delta G. Non-catalyzed basically means without an enzyme, right? Catalyzed means with the enzyme, right? Because an enzyme's a catalyst, right? So with the enzyme, catalyzed, without the enzyme, non-catalyzed. Okay, got the diagram? Pretty easy? Okay. All right, and then ATP, ADP cycles. So ADP, okay, so energy from catabolism. Catabolism means what? Breaking down, digestion, right? That gives you the energy to do what's ADP. Make it into ATP, right? So then it becomes ATP, and then here you use it for your cells, right? And then once you use it for your cells, it's now become yeah, let's just make this ATP, and then it becomes ADP plus P, and then the energy from food makes it again, and then you use it for your cells, whatever, and then there it is. Got it?
No.
What kind of, no? Okay.
We can't see.
I cannot do that.
All right, I'll do it again. Okay. Energy from catabolism. Okay, so here we have ADP plus P. Okay, so the energy from our food goes into the mitochondria and makes ATP, right? What is ATP used for in your body? Cells. I mean, that's a very broad word. Muscle contraction, active transport, even the act of thinking uses ATP, okay? And your brain, the energy your brain needs the most, that works the best, is sugar. That's why when you study, you crave sweet things, because your brain wants more sugar to get you thinking, okay? And making more ATP, right? And then cells use the ATP, and that ATP is broken down into ADP and P, and then how do you make more ADP and P? You eat food and breathe. Is that a better diagram?
Yeah.
Okay.
Don't embrace it.
Okay, you ready? Next chapter? You guys hanging in there? Sleeping on me? Case is. Okay, here we go. So let's write the equation. Not that you have to know the equation, but you have to know how to interpret the equation. Okay? So what is it?
6H12O6 plus O2. And then arrow. 6CO2 plus or something. 6H2O plus.
Yep. Okay, so products and reactants, right? So we got the reactant side, we got the product side. Let's talk about oxidized and all that stuff. So what ultimately happens to glucose? It is oxidized into CO2, right? So the food you eat, okay, is broken down and essentially is exhaled as CO2 and given off as water. So you sweat it out, you pee it out, right? But this is oxidized. I'll put oxy. Oxidation means what? Breaking down, rust, oxidation, break down the rust. The car is old, the car is rusty, right? What happens to oxygen? It is reduced into water. Reduced meaning to build up, okay? Yeah, I know, it's chemistry, the nature of chemistry, so. Okay, so since I have that up there, let's talk about stages. So what stage does glycolysis, what stage I can't talk. It's glycolysis, right? Glucose is initially entering in glycolysis, right? What stage does oxygen play a role? ETC. Okay. What stage does CO2 released? Krebs. Okay. And then what stage do we make oxygen? ETC.
That's water.
Water. Okay, yeah. Water is made during the ETC. Told you. I'm hazy. But yeah, those are right there. Okay? Okay, so products and reactants, check. Oxidize, reduce, check. Inputs and outputs. Okay. So on this, it's the main things. All right? So let's look at glycolysis. Let's look at in, and then over here, we'll say out. Okay? So what goes in? Glucose. Okay, so glucose goes in. You also need an initial input of what? You need an initial input of 2 ATPs. And you need an investment to get the process started, right? Okay, so that's what goes in. What's made?
2 ATP.
Yeah, 2 pyruvates. What else? 2 ATP and 2 NADH. Electron carriers, right? Those are carrying the hydrogens. So where is glycolysis? Cytoplasm. Okay? So the unique thing about glycolysis is it can happen with or without oxygen. It can be aerobic or anaerobic, all right? So let's go ahead. Do you want me to do the anaerobic with it or jump to, or just do all aerobic first? What'd you guys say? All together? Okay, so we'll do aerobic first. Okay, so let's say oxygen's available, right? So oxygen's available. We made the pyruvates. These 2 things. This is going to play a role later, right? Where is this going to go? In the piggy bank, but what stage will we eventually use it? The ETC. So right now, it's in the bank, and we're just kind of holding on to it. technically you make 4 ATPs,
But two have to go.
but
back.
2 have to go back, right? So the net gain is always 2, okay? So I'm gonna put net, right? Okay, so because oxygen's available, which one of these things moves on? Pyruvate, okay? So we have in, out. Okay, so pyruvates go in. Okay, so again, 2 pyruvates. Technically, it's 1 pyruvate at a time, but we're just going to put pyruvate. Okay, so pyruvate enters, and we have that in-between stage, right? It's not really the Krebs, so we're just going to take pyruvate and make it into the other thing. What is that other thing called? COA. Okay. So, but as for knowing like the amount of CO2 and all that in the in-between, I want you to know the grand total for the whole process. That's what matters. Okay. So acetyl COA is made from pyruvate. Then we go through the reaction. Okay. By the time we're done, okay, all this is gone, and what gas is given off? Yep, 6 CO2. And you just exhale that out of your body, okay? But we make a lot of these. What are they and how much?
ATP.
Okay, yeah, NADH. Sorry, I was thinking NADH first. And what's the other one?
ATP.
FADH. Okay, and how many ATPs? 2.
Is it 6 or 8 NADH?
You make 2 here in this in-between stage and 6 in the Krebs, but let's just do the total. It's 8, yeah. So we have 8, okay? So, and again, just like over here, these 2 things are going to go where in the end? To the ETC, okay? So far we've made 2 ATPs in glycolysis, 2 ATPs in the Krebs. We've completely oxidized glucose into CO2. You've exhaled it. Okay. What happens in the ETC then?
NADH and FADH.
Right. So this NADH and these NADHs and FADHs go here.
Yeah.
So I'm going to do the total. 10 NADHs and 2 FADH2s enter. Okay. What else enters that we haven't talked about?
The oxygen.
Yes, right there. Okay. Oxygen enters. Oxygen's always forgotten about. Okay. So then so when NADH and NADH, so here's the membrane. Okay. When they reach the membrane, what happens to them? The H's are stripped off and they go to the intermembrane space. So then NADH becomes NAD and FADH becomes FAD, and there's all these H's. They're also called high energy protons, right? Because they have a positive charge. Okay, so NADH and FADH are stripped, right? You can't really say they go out. They're just broken down into NAD and FAD. Okay. What happens to oxygen?
Bonds with hydrogen.
It bonds with hydrogen. So again, we have all these hydrogens that are going to pass back through, right, with an electron. And so these oxygens,
H2O.
okay, with a negative electron and hydrogens are going to make 6H2O. So what's the last electron acceptor? Oxygen is the last electron acceptor, forming water. Okay? How many ATPs did we make?
26 to 28.
26, 28. Okay? For a grand total of 30 to 32 ATP from one glucose molecule, right? One single glucose. So if you had Fruity Pebbles this morning and a slice of toast, you just ate a ton of glucose, you're making bazillions of ATPs. Okay. Okay. How you feel about that stuff? Now let's go back. Where's the Krebs? This is the matrix. And where's the ETC?
Outer MCM.
Inner
Space.
membrane, also called the cristae, right? Cristae. I think someone should name their child Cristae instead of Christy. Okay, so how you feel about that? Okay. And you know, it's a pathway. So if you like make sense of the math, it just kind of, it makes logical sense. All right, let's finish this up. Did that, we did locations, we did what happened, difference between substrate level and oxidated. Okay. Which two processes makes ATP using substrate level phosphorylation? Okay. Substrate level phosphorylation, making ATP with an enzyme. Okay. Oxidative phosphorylation is the ETC. Oxidative phosphorylation is hydrogen ions passing through ATP synthase to make ATP. It's the oxidation part. These are oxidized, right? Because they lost their hydrogens. That's the oxidative. The phosphorylation is making the ATP from ADP. Got it? What'd you say, Al? Okay. So that's the difference. Making ATP with an enzyme and then moving hydrogen ions. Okay. Oxidative phosphorylation is also called chemiosmosis, but I didn't really put that on there, so you don't really have to know that per se. Okay. Know the role of hydrogen ions. Okay. What do the hydrogen ions do? Yeah, they pass through ATP synthase, oxidative phosphorylation to generate ATP. Okay, where are they found? This is the inner membrane. This is the outer membrane. What's this? This is the intermembrane space, the space between the inner and outer, right? And they're going to diffuse through to make ATP synthase. Okay? What'd you say? So they make ATP where they found the intermembrane space.
Oh my gosh.
What?
So where do they diffuse through?
ATP synthase. This that thing that spins that.
Oh yeah.
Okay. So here's the inner membrane, the outer membrane. This region is the intermembrane space. Good on that?
No.
Okay, ask me a question.
What's the rule? Can you redo it?
The role of hydrogen is to make ATP.
That's it?
Yes, because what do they move through? ATP synthase. And when they do that, that spins and that generates, converts the ADP into ATP. The windmill. Yep. Or the mill on a river.
So it
The
doesn't,
road, the wheel.
it doesn't matter that they're carried by FAD and NAD?
Not in this case, not in that particular one. Okay. What's unique about glycolysis? It can be with O2 or without O2, right? All of life does glycolysis. It's the oldest process in all of life because all of life makes ATP, even though not everything has mitochondria. Okay. All right. And then the last part's fermentation. Can I clear this? It's getting kind of. Okay. All right, fermentation. So tying in with glycolysis, because glycolysis only does fermentation, because glycolysis is anaerobic, right? Two types of fermentation. Lactic acid and alcohol. Okay? All right. So, again, what do you always start with in glycolysis? Pyruvate. Okay? If it's an animal, pyruvate is going to be made into lactic acid. Okay? You make NADH along the way, but you don't put it in the bank. You immediately use the H because that H becomes part of the compound that makes lactic acid. Okay? Why do you make lactic acid? Because you're not getting what in your muscles? You're not getting oxygen. It's a way to make ATP. So you make lactic acid and a little bit of ATP, enough to move your body. Okay? Now, if it's a plant like a potato and we want to ferment it, okay, we're going to take the plant, we're going to take pyruvate and make NADH, but immediately strip it and make it into NAD, and we're going to make an alcohol. What's the name of the alcohol? Ethanol. Okay, but ethanol is a 2-carbon compound. This is a 3. Okay? So what's the gas?
Two CO2.
Yeah. So that's our 1 carbon. There's our 3, because remember this is a 3 carbon. So you make a 2-carbon ethanol, a 1 carbon CO2. Why is beer foamy? CO2. Why does bread rise? CO2, right? The yeast gives off CO2, the bread rises, and then the waste product is the ethyl alcohol or ethanol. Okay. Glycolysis, no oxygen. That means that's 1 carbon, 2 carbons
What's the blue arrow for?
given off. Right. You open that can of root beer and pour it, it's all foamy. That's the CO2. So, yeah.
Is lactic acid a type of alcohol?
Lactate, no.
Okay.
No.
Never mind. I was gonna ask if you can drink it, but.
Yeah. Fun fact, it's not why your muscles get sore. Your muscles are sore from micro tears from lifting. But if it's present, you typically have micro tears. So. Okay. That's it. Did you survive it? Did you